Okay guys, grab a seat. I'd like to introduce our next speaker. It's Dr. Judy Carman. Judy's been, it's the second time to Rockhampton to speak for us. Last year she came along and spoke as part of the discussion with Don Huber, and there was a string of speakers last year on agricultural solutions and also human health. And that was a pretty dynamic 2 days that I missed most of. But if you want to have a listen to what Judy said last year, it's actually on our Healthy Soils website, or you can just get a memory stick from us, a thumb drive with all of the presentations and discussions. So Judy is she's got more qualifications than I can say. She's an epidemiologist. She's got multiple degrees and in so many areas that allow her to be able to, actually I'll let you say what you've got, that'll allow her to be able to run research trials with other teams of scientists where they can actually assess the safety impact of genetically modified organisms. So this is probably the most qualified scientist in Australia to do the work that she's doing. So without going any further, Judy.
Thanks everyone. First of all, a sound test. Can you actually hear me? Everything all right? Okay.
Can't see you.
Can't see me. I'm going to try and stand here a little bit more so people can actually see me. So I'll just go through my qualifications first because they are pertinent to this. So I've got an honours degree in organic chemistry, so I used to make chemicals. I've got a PhD in medicine in the field of nutritional biochemistry and metabolic regulation. A lot of animal feeding studies in that. I've also got a Master of Public Health specialising in epidemiology and biostatistics. I taught at Roseworthy Agricultural College for 3 years. I've worked in HIV/AIDS, I've worked in nutrition at CSIRO. I also was the senior epidemiologist in the Communicable Disease Control Branch of the South Australian Department of Health, investigating outbreaks of disease, most of which were foodborne. And as part of that, I also did the epidemiological investigation into whether rabbit calicivirus could infect people after it escaped from Wardang Island off the coast of South Australia and spread through the country like wildfire. The government decided after it had escaped, maybe we'd better see if it's actually safe for people to be exposed to it. And so I was called in and did the multi-state study into whether it was or not. And I'm happy to report that you can eat rabbits that have got rabbit calicivirus in them. There was nothing to be worried about there. And in addition to that, I've been on a member of ARTEC, which is the ad hoc technical expert group under the Cartagena Protocol, where risk assessment documents are being developed for countries to use. So this is a United Nations committee that I've been on. So let's start. Now, how do I turn the...
Do you want me to do it or you do it?
If you want to, yep. I've got the critter.
Sure, push that back in.
Right. I've got a mouse. Right, so just a quick recap of the sort of things that have already been said before. There are different ways of making GMOs, one of which is the biolistics method. But one of the main problems about this is that the genes are inserted randomly. There's a question about whether it might affect the functioning of the plant, whether new substances might be produced as a result. Bits of DNA can go in there from a variety of different organisms, including other plants, animals, bacteria, fungi, viruses. There's already been a discussion about cauliflower mosaic virus and the fact that antibiotic resistance marker genes are often in there. There are two main classes of GM crops. First one is that they're made to be resistant to a herbicide, and the most common one there is glyphosate Roundup. And the other type makes its own pesticides. In many crops, particularly we're talking about things like corn, cotton, corn, it's called corn in the United States, we would call it maize here. Corn, soy, cotton and canola are the main ones still that are genetically modified. There's an awful lot of multi-stacking occurring, which is why I was asking the questions of Renee about what is actually currently in the cotton crops that are being grown here. So in that case, there was a stacking of 4 different genes in there, which is herbicide tolerance 2BT 5. And they're the two main types still. So we've got herbicide tolerance and we've got insecticidal proteins. So the aim is you put DNA, it's traditional GMing, is to put a piece of DNA into the crop so that the crop now produces new proteins. And those proteins are the things that provide the certain characteristics that are bred into these plants, and they're particularly to be resistant to a herbicide. So that now you can spray the crop with the herbicide, the crop will live and the weeds around it will die. And the other one is to make an insecticide. So the plant now produces an insecticide, so when a particular grub comes along and eats the plant, the grub will die. And there's a lot of multi-stacking, as I said. We're getting up to 8 stack in corn in the United States. So let's look at the safety testing, the gold standard. So what I'm going to do is look at the safety testing of GM crops and compare it to the gold standard. So let's look at the gold standard. Now this has been developed through pharmaceutical drugs, which have had some good news reports and some bad news reports. And as a result of that, there is now a procedure that is used in pharmaceutical drugs. The first thing is you make your pharmaceutical drug, your new one, got great ideas of how it's going to work. You test it in animals first and to see if there's any deaths or if there's any bad effects on the animals. If it passes that stage, you then go into starting off in clinical trials in people, where you start off safety first. Does it cause toxicity to people? Small numbers of people at this stage. If it passes that, you go to phase 2. Does it have a beneficial effect you're after? If it passes that, you go into randomised controlled trials, which is where you split people into one group gets the drug, one group gets a placebo or the standard treatment, and you compare, how many people get sick, how many people get better, what the side effects might be. Now after that, there's also monitoring them in the community, so that you can see, because when you do pick people for a clinical trial, you do things like pick people who are healthy. Generally, you don't want people who are pregnant, so therefore, does the drug have an effect on pregnant women? You don't know because you've screened out the pregnant women. So therefore, you monitor in the community to see if there's now an effect on pregnant women that becomes glaringly obvious. There's also now been a further stage that's been often included, and that is what's called a meta-analysis, where all of these randomised controlled trials get put together and one understanding comes out of it, a relative risk or a relative benefit as one number when you pull all of these trials together. So let's now compare what happens with GM crops and how far through this process GM crops have come to understand just how thorough the safety assessments of them are. And unfortunately, there are no clinical trials. All of this from here down is just not done on GM crops. And I argue, and you'll see my arguments here, that even the animal testing that's done, if it's done, is actually woefully inadequate. So how did I come to this conclusion? Well, I looked at the evidence. So I went through Food Standards Australia New Zealand, which is the regulator not only for Australia but for New Zealand, and I looked at their reports. So I reviewed 12 Fazant's reports covering 28 GM crops. So first thing you'll notice is that they will come to an assessment sometimes on not just one crop per report, but several. And I looked at what do they need from the company to determine it's safe. What do they actually do? How does it compare to the gold standard? Now, there are 4 main concerns in consumers' minds and in the public health mind, in people like myself who've been trained in public health. The first one, does it cause allergic reactions? Does the GM crop cause an allergy? Does it cause cancer? Does it cause reproductive problems? And does it cause a toxic result, toxic outcome? The allergies are only done theoretically. They are not tested in animals for allergies. So this is, for example, we've put this gene into the plant. We expect that protein to come out from it, and we're going to assume that nothing else happens. No other proteins get produced. We're now going to look at that protein. We're going to artificially chop it up on a computer, and we're going to compare that to known sequences of known allergens that are in the environment, like shrimp and, cockroach allergy, the things that might cause cockroach allergy. Sorry?
Peanuts?
Yes. And we're going to compare, but this is done on a computer, not on animals, and it's only done on a subset of all the allergens that we know about, all the allergens that there are. Animals are not fed for long enough for cancers to develop, so there are no cancer assessments. Reproductive, not done, and then there's toxicity testing. Now, some of that is done, and I'm now going to tell you exactly what is done by Fazants. The first thing is that generally what Fazants requires, what they require, is simply a compositional comparison. So they'll get some samples of the GM crop, some samples of the non-GM crop, and they will look at usually only the amino acids as the components that they're looking at. So they don't look at proteins. They break the proteins apart into the building blocks, and they just look at the amino acids. Now this is a little bit like comparing the fire standards in this building and maybe the one next door, not by looking for the fire exits or by looking at the stairs or the alarm systems, but by knocking the building down and counting the bricks. You've lost all the information that you actually need to determine if something's an allergen, right? You've broken it down into the amino acids, because it's pretty hard to get an allergy to an amino acid, but a protein's a different thing. Sometimes they might look at anti-nutrients, so this might be things like erucic acid or trypsin inhibitor in soy, erucic acid in canola. They have very small sample sizes. Now this is important because if you don't want to find a difference between two separate groups and you have small sample sizes, that's really easy to find. It becomes far harder to find, or far easier to find a difference between two groups if you increase the sample size. So here we're talking about sample size of 5 to 7, and sometimes I've seen a sample size as low as 1 or 2. There's insufficient statistical information. So normally in a peer-reviewed scientific journal you would expect certain things. You'd expect the mean, the standard deviation, the confidence interval of the test, the name of the statistical test, the p-value. These, in the ones that I looked at from Fazants, they were not there. Some of them might be there sometimes, but on no occasion did I see all of them there as they should be. In terms of the definition of substantial equivalence or compositional comparison, what Fazants then says is it's substantially equivalent. The GM crop is substantially equivalent to the non-GM crop, but there is no definition about what that means. So it's a bit like not knowing, if there's no speed limit, how can you fail the speed limit? So with no definition of what constitutes compositional comparison, everything passes, including, in fact, in one corn, which is one of the corns that's been talked about here before, 44% of the amino acids were statistically significantly different, even on really small sample sizes, and Fazants still said that it's substantially equivalent and it's safe to eat. And that one did not have any animal studies done on it at all. So Fazants does not need animal studies, does not need any at all in order to determine that it's safe to eat. Certainly does not need human studies and does not review the raw data. So when a company like Monsanto provides information to Fazants, they will say, here's raw data, here is our summary of what we say the raw data says. Fazants looks at the summary of what Monsanto says is there and only looks at that. They have a policy of not looking at the raw data to determine whether what that GM company says is in their raw data is true or not. And there's even less for crops that have got these stacked genes. Essentially, there is an assumption that if Fazants has approved that gene in that plant, and that gene in that plant, and that gene in that plant, you can put them all into the plant, and there are no more safety assessments required. In other words, there's this assumption that they're not going to interact. Now, we know with pharmaceutical drugs that there are interactions between the things that you might take, and that therefore, if you're going to, in pharmaceutical drugs, if you put two drugs into the same pill, you actually have to do safety testing that there will be no interactions, or to at least know what the side effects are, but that doesn't occur with GM crops. And these stacked crops are very common now, and particularly in the United States. So 3/4 of US corn planted in 2014 was stacked with both herbicide tolerant and BT genes. Now I was talking about they do some toxicity studies. So let's have a look at some of them. So once again, Fazants does not require these to be done. But these are the sort of toxicology studies that the industry does on a GM crop. And it was of the ones that I reviewed that Fazanz had said was safe. It was the only testing done for 61% of them. There were no other toxicity studies done. What they do is they say, okay, we've put this gene into this plant. We now expect that there will be this protein produced. We are going to take that protein and we're going to put it down the throats of some rats as one single dose, and we're going to see if the rats die. And this assumes quite a few things, and they're only going to observe for 7 to 14 days. Now this assumes quite a few things. First of all, that GM crop is only going to produce that protein and nothing else will happen. And as we've seen, there might be other things that might actually be being produced because of the way that they're actually inserted. There's also the assumption that the protein that they've tested on the rats will be exactly the same as the protein that the plant produces, except the protein that they put down the throats of the rats hasn't come from the plant, it's come from bacteria. So they've spliced it into bacteria producing that protein. But the problem is bacteria are more primitive critters than the plant. So their plants, once they make the protein, can alter it. They can put sugar units on it. They can make other adjustments afterwards or other changes that bacteria can't. So the plant produced protein may not be the same as the bacterial produced protein. And the other big ask is that it will create disease within 14 days. Now, there are an awful lot of things that are really quite toxic to people that if you gave people an oral gavage would come out as a clean bill of health. Smoking, alcohol consumption, lots of things. Now, there is a better quality of standard, and that is to actually you get the crop, you feed it to the animal, and you feed it for long enough to see if things are going to happen. If they do this, there's no requirement on them doing this, but if they do this, it's standard procedure for the company that if it's a herbicide-tolerant plant, they do not spray it beforehand. So it will come into the food supply of people sprayed because that's what it's designed to be, right? But it's not actually sprayed when they feed it to animals. They also use what I would regard as unusual human health models. And that is, they tend to use farm animals a lot that are not physiologically comparable to humans. So they might use things like chickens, cows, and trout. Now, chickens are physiologically very different to people. You know, we have kidneys. They don't. They've got this strange smear along the backbone. We have one stomach, not two. We don't swallow grit to help grind our food. We also don't have feathers, we don't lay eggs, you know. So there's a lot of difference between a chicken and a human being, which is why they're not used in medical research. And similarly with fish, I mean, we're definitely not like fish. Then the endpoints tend not to be terribly relevant to human health. So for example, they'll feed it to some chickens and they'll measure breast meat yield. Because obviously that's important to a farmer, to know that if you feed that crop to your chickens will live for long enough and get a good price at market. But that's not a measure of human health. Otherwise, sorry men, we're not very healthy compared to women. So it's just not completely relevant, is it, to human beings. Or they'll measure things like abdominal fat, weight, with the view that if your abdominal fat had weight, the healthier you are. These are actually animal production studies. So the actually will feed animals for 3 months. But in the ones that I looked at, the ones that I reviewed, this was very uncommon. Now it's becoming a requirement now in the EU that GM crops to come into the EU need to go through a 90-day toxicology study, which is still not a long-term study, I hasten to add. That's more like 6 months up to 2 years. But once again, animals are not being fed long enough for adverse events to develop. So the number of animals in these sorts of studies are often really quite low. And this becomes important because if you've only got 20 rats in a group, and if your end point is do they die or not, which I think you will agree is a pretty rough understanding of whether something's healthy or not. Because we all know people who are ill, but they're not dead yet. You know, people with cancer, with diabetes, with all sorts of health problems. If you're only looking at death as an endpoint, which is often what they do in their studies, then anyone who is not dead yet is apparently healthy. Now, if you've only got small numbers of animals, because we're starting to talk about rat studies here, if you've only got 20 rats in a group, then, and you're looking at death, then the way the statistics falls is that a quarter of the animals have got to die in one group and none in the other before you reach statistical significance. If it doesn't reach statistical significance, everything's fine. If you've only got 10 rats in a group, half the rats have got to die now before you reach statistical significance. So if you've got 20 rats in a group and only, 20% die, everything's fine. Because if something's not statistically significant, it doesn't exist in science. Once again, often only body weights and death rates are used, with the aim, of course, that the bigger the body weight of the animal, the healthier it is. Now, I would suggest to us as human beings that we probably don't want to put on any more weight. And yet this is regarded as a good thing in an animal study. If they go further than that and actually do things like look at organs, generally what they do is they will take the organs out and weigh them. They don't look inside. And you can have a diseased organ and a healthy organ and they can weigh the same. So the organs are rarely internally inspected. There's generally no blood biochemistry, and certainly there was none done in all of the ones that I looked at. And if there are adverse effects found, and there are, that's not investigated further. So, for example, rat-up-ready canola, which is one of the big hot topics in farming systems in Australia today, when that was tested on rats, there are a few things that are important here. First of all, you and I eat the oil. It was not tested. So the bit that humans eat was not tested. The bit that was tested was the bit that was left over once you crush these seeds of the oil. Right? So that was the canola meal, that was fed to rats. They had an increase in liver weight of 12 to 16%, and it was ignored. It was not investigated further. So on the basis of these sorts of tests, lots of things that we know are harmful to people would be given a tick of approval. Tobacco, asbestos, ethanol, thalidomide, because there are no reproductive studies, chronic metal exposure, etc. Now in terms of, and I should say that this was been published. Well, everything that I'm talking about here is published research. So in terms of labelling, because the labelling issue has actually come up here, and I haven't actually got a specific slide on labelling, there's been this view that you can trust your labelling laws, but there's an awful lot of things that are not labelled. Anything that is fed to an animal and the product comes from the animal, that doesn't have a label. So if you feed your cows and the cows get milked and the milk goes in the supermarket, that's not labelled, because there's a view by FSANZ that it's not possible to find GM DNA or protein in milk. However, there was a study done in Italy where, trying to look for the actual number here, a third of the milk samples from supermarket shelves were found to have GM DNA in them, and in fact they could tell which GM crops the cows had been eating by looking in their milk. There's also a view that oils don't need to be labelled because apparently there's no protein or DNA in oils. That's not the case, I'm sorry, but it's not, and it's well understood in the scientific literature and the nutritional literature that there is protein in oils, and that's been known for a very long time, because the oils that come into the food supply are not that well purified, not to an analytical reagent grade that you might get in a laboratory. And so, for example, there was, so in the peer-reviewed scientific literature, once again, there is an understanding on evidence that about 22% of people who are allergic to peanuts can't have peanut oil. And there has in fact been in South Australia, there was a few years ago a very celebrated case in the coroner of a woman who went to a restaurant and said, I'm allergic to peanuts. Do you have any peanut oil in this food? She was told no. She ate the food, it had peanut oil in it. she had an anaphylactic reaction and she died, and the coroner quite specifically said it was the peanut oil that she ate. As soon as you go outside your front door to eat at a restaurant or a bakery or a takeaway, does not have to be labelled. Anything that's unintentionally contaminated to 1% per ingredient does not need to be labelled. If you've got a bacterium that is producing a food flavouring, that does not need to be labelled. And so there's a great deal that's not labelled, and that concerns consumers about what is and what isn't labelled. So they become wary and upset about the GM foods. People generally want to have a choice. They want to have a choice whether they eat this or not, and they get very concerned when they're not given a choice. Now, with the labelling requirements that I've said and the safety assessments that I've described, things are worse in the United States. There is a view in some places in Australia that the FDA has approved these GM crops for human consumption. That's not the case. It's a voluntary system over there, so a GM company, when they go to the FDA in the States, doesn't have to go to the FDA. They can simply put it on the marketplace. Generally speaking, however, they do provide information to the FDA, but at the end of that process, the FDA does not say this is safe. What they say is, thank you very much for your information. We understand you, the company says it's safe, and we have no more questions. So I'm now going to move into histopathology studies, because we also did a review of these types of studies. Now, if you're going to see if there's any effect of a GM crop on an animal, the best way is to actually look down a microscope. So you feed the animal for a period of time, and you look down a microscope to see whether there's any damage to the organs. So you don't weigh the organ, you go further than that. Cutting the organ open and looking at it like a kidney, you go further than that. You take slices out, you look at them under a microscope. So we did a review of the histopathology studies that had been done, and we limited it to, the reason will become obvious a bit later on, to 3 particular GM genes, and this was the herbicide tolerant trait and 2 particular BT proteins. So these are GM crops that have these, so they might have one of the genes, they might have 2 of them, they might have all 3 of them. And we looked at just rat studies that have been fed for 3 months, and we also just looked at the digestive tract, because that's where you swallow it. You chew it up, it ends up here. So this is the first organ it comes in contact with: the stomach, the small intestine, the large intestine. And we had a look to see what's been done, what safety assessments have been done. So we did 47, looked at 47, we found that there were 47 crop varieties that had 1, 2 or 3 of these genes in them. But we also found that 76% of these, there were no published studies for 81% of those crops. None. No safety assessments on histopathology at all. And of the ones that were done, 76% of them were done after approval, with half of them at least 9 years after approval. So clearly, these studies were not available to the industry, not available to regulators when the regulators decided that they were safe. What we found was that there wasn't a single study properly conducted or reported. So, for example, they weren't clear about what they did. Their methods weren't clear. They did things such as say, we took samples from the stomach, the small intestine and the colon, but then they only report results for 2 of those and not all 3. So where's the 3rd? What's happened there? Some said they didn't even tell you which organs they looked at. Some won't tell you what histopathology methods they used. Some will say, we didn't find anything adverse, without telling you what is adverse, what in their opinion is adverse. So once again, it's rather hard to be pulled up for speeding if there's no speed limit, and if you're not clear about what the speed limit is. So, and some saw changes and then just dismissed them, okay, and didn't go any further. So we decided that there was actually a lack of evidence that these crops were safe to eat. Now I'm quickly going to divert to some new techniques that are coming through now, because to date we've been talking about the standard sort of GM crops. There are two new that I'll talk about just as an introduction, if you like, of some new ones that are coming through. One's, I call it the CRISPR type method. Now these are also been called GM 2.0. Now there's a big debate that's happening in Australia at the moment about whether these should be regulated at all or not. And if so, what sort of regulation, when do you start kicking in the regulation? When that kind of change has been made, or a bigger change, or a bigger change? And so there's a big debate going on. What the industry would like to do is to deregulate all these CRISPR type of processes, as well as the double-stranded RNA, which I'll get onto very shortly. They're saying that it's more accurate than the previous methods. I'm going, hold on, didn't you say the last methods were accurate? How can you be more accurate than accurate? And in fact, with the results coming through now, it would appear that they're not terribly accurate. You can get quite large deletions at the site of insertion. These work slightly differently. Basically, they do a seek and destroy, if you like. So they go and try and find a particular spot in the DNA in the CRISPR method, and they try and cut the DNA at a particular spot, and then they may or may not actually insert new DNA into it. The problem with this is there may be problems in actually directing it to the right place. So you need to have enough information for that piece to go there and not there and there and there and there as well. And it would appear that that's not terribly accurate. Now with the double-stranded RNA technology, this is, and I should say, so it looks like there are unintended effects. The Austrian government is one of the few governments to consider the risks, and they've decided there's insufficient knowledge about the risks and it should be regulated like all others. There's a legal decision made in Europe just recently that these should be regarded as the same as ordinary GM techniques. So let's have a look now at double-stranded RNA, because this is a new technique that's coming through now, and the CSIRO, for example, has patents on it, fundamental patents on double-stranded RNA technology. And it's coming to a paddock near you, and it's coming into our food supply. So the aim here is, instead of the plant now producing a new protein, the aim is to go and silence a gene, usually to actually silence a gene or similar things to that. So what's happening is the CSIRO, for example, is trying to change the type of starch in wheat by silencing a branching enzyme. In insects, it's being used so that the plant will produce this double-stranded RNA. The grub will come along and eat the plant. The double-stranded RNA will go into the gut of the grub. It will be absorbed into the tissues of the animal. It will silence a gene in the insect, sorry, not an animal, in the insect. It will silence a gene in the insect, and the insect will die. There's also moves to have sprays, which are double-stranded RNA sprays, that can be sprayed from planes or knapsack or whatever, but certainly including from planes. Now, there was a view that, of course, this wasn't able to change the genetic expression of people, except once again, you wait for the evidence to come through, and then you tend to find information being presented in the peer-reviewed scientific literature that things may not be as rosy or as clean as you would like. And certainly there is work, and this has all been published once again in a review, that double-stranded RNA in even non-GM plants can actually survive digestion and actually influence your gene expression. And this has been done in rice. It's also been known that you can change gene expression in mice and in human tissues in tissue culture. So we actually really need to go through a proper risk assessment of this, I would argue. So in the review that we did, we looked at the peer-reviewed scientific evidence for safety, and then we looked at 3 government regulators to see, are they looking at this? How are they looking at it thoroughly? Are they looking at the peer-reviewed scientific literature to do a proper safety assessment on them? And of the 3 government regulators, we found that safety was not considered at all. Or if it was considered, it was assumed that it was safe. In other words, they weren't going into the peer-reviewed scientific literature to see if it was safe. And they weren't requiring evidence that it was safe. So the plan for the CSIRO wheat, for example, This is a means of silencing a branching enzyme in the wheat, so that now the wheat, instead of having branches in the glucose molecules that make the starch, will be now mostly straight lines. The aim being that when you eat it, it takes longer to digest, so there might be some more of the starch that enters up in your large bowel, and that therefore that might actually improve your chances of producing bowel cancer and things, resistive starch. The only problem there is that we have a branching enzyme that is rather similar. There was no view that maybe they should see if it might also inhibit our branching enzyme. They were going to do some human studies, but it was only going to look for the benefits. They weren't even going to consider looking for any adverse effects. And there are some people who are born without this branching enzyme, and they tend to die before the age of 5. So clearly, if it does inhibit our branching enzyme, it could be quite catastrophic. So maybe they'd better go and actually check first. So there's the published publication here. And we've come up also with the way that we believe that it should be safety assessed, and that is, first of all, to determine if there are any actually likely unintended effects from that double-stranded RNA. Check first. Identify all of the ones you expect to find in the plant and any unintended ones as well. Test on animal and human cells in tissue culture, do some testing on animals, and then if you intend for people to eat it, then test on people as well. And it should include things such as, with the wheat, for example, that they were talking about. Bakers bake with it, and people at home, you can inhale the dust. So it might be a good idea to check routes of inhalation, as well as dermal contact, as well as eating contact. So, in terms of the regulation, there are a number of new techniques coming through. Things are rapidly evolving and changing. Some of these techniques are definitely in their infancy. Some of them are being developed as we're speaking right now. And it's my view that they really should go through a proper risk safety assessment before they come into the human food supply, before farmers get exposed to them, and before people eat them. And if there's no regulation, and there is a push by the industry to have this all completely deregulated, no regulation means no safety testing, no labelling, you'll have no choice, you'll be exposed to it whether you want to or not. And it even includes things such as, there are kits you can buy on the internet now where you can set up your own, gene editing at home. And there will be no regulation of these either. You could get a microbrewery down the street who might buy one of these kits, might alter the yeast, might then start brewing beer and selling it to people, and it's not gone through any safety assessment at all. In addition to all of this, I should point out, the industry is saying the same sort of thing that Jonathan Latham was saying. The industry is saying certain things to the regulator, but if you look at the patents, you see something else. So what the industry is saying to the regulator is, this double-stranded RNA, it's not heritable. It's not possible that it can go into the germplasm. So you can put it on, you can spray it on your crop, and it won't go into the seeds. And certainly, if you eat it, it won't go into your DNA. But the patents say something otherwise. The patents clearly say that Monsanto, for example, claims that when they can spray a crop with the double-stranded RNA, they claim the crop's theirs. If they spray a forest once, and the forest lasts for 50 years, that forest now belongs to Monsanto. If there's overspray onto a neighbour's field, that belongs to Monsanto as well. But more than that, they also claim in these patents, which have been granted, that their progeny, they own their progeny. So it's the seeds, etc. They can claim those as well. So that's labelling. So let's go to there were reviews and stuff that we've done and published in peer-reviewed scientific journals. Now I'm going to go to a couple of studies that we've done where we actually go, well, what happens when you exceed these standards? These woeful standards for safety assessments that I described before, where you don't need to do any toxicology study, you don't need to do any long-term studies. So let's look at that. So why pigs? Why did we do it in pigs? This was done in the States, I should say. Because pigs are like us. Their digestive system is the closest, much closer than a rat, certainly closer than a chicken. We've got a similar digestive system. And it was also because one of the co-authors of this paper, Howard Vleger in Iowa, was saying that he was a farm advisor on crops and on livestock, and he was saying, look, we're seeing problems in the States. We've been feeding GMOs to our animals for quite a few years now. We're seeing problems, particularly in pigs, reproductive problems such that the sows, we're finding it harder to conceive, get the sows to conceive, and there are higher rates of miscarriage. And generally speaking now, there's so much artificial insemination occurring that it's hard to work out what the effects on the male reproductive system might be in pigs. But being the United States, there are some communities that, such as the Mennonites and some religious communities like the Amish, that actually require the boar to be mated to the sow. And there, they actually find there's a reduction in the piglets born if boars are used instead of artificial insemination. Howard said, look, we're also seeing problems in the digestive system. We're seeing inflammation in the stomach and then in the small intestine. And it can get so bad in some of these pigs that they can literally go down in a screaming heap, so that they will literally bleed out. And this is particularly in the ileum of the small intestine. They'll just rupture and bleed. So we used standard, this was done in a commercial facility. So we used commercial Yorkshire Cross standard, got them just weaned, all iso-weaned, so they're all the same age, or weaned at the same time. The boys were neutered at 3 days of age to prevent boar taint, so we don't know what's happening in the male reproductive system. That's important to note. We had a nice sample size now. We've got 84 pigs in a group, not 20, not 10, but we've got 84. So we've got a good sample size. We fed them for about 5 months, the complete commercial lifespan of pigs. So now we can see maybe some longer term effects. We've got equal numbers of males and females. Each pig was individually identified so we could follow them through, and we monitored a lot of things on a daily basis: the food intake, the temperature, the air quality, any skin problems, any eye problems, have they got any diarrhoea, all this sort of stuff. And this is what it looks like. So these are little piglets arriving in a school bus, a modified school bus. And then they're randomised into, so this is just after they've arrived, and they're randomised into pens. And then this is the latest stage. And I don't know if any of you have raised pigs, but they get big. So they get over 100 kilos, some of these. So this is the finishing stage. So the feed. We fed them as they're fed in the States. And in the States, they get fed soy and corn. So we fed a mixture of these as according to normal US pig industry standards, and they had the three genes that we reviewed earlier. So they've got the herbicide tolerance, and they've got those 2 same BT genes in them. They were split randomly, completely randomised. One half was fed the GM diet, and the other half was fed a non-GM equivalent diet. Now, there was no isogenic control available because the triple stack corn does not have an isogenic parent. But they were all processed on the same equipment, so we had the same grind size. We had everything was exactly the same in making the GM diet and in making the non-GM diet. We also measured lots of mycotoxins, and they were well below the EU level of concern. And there was also bentonite clay added in there, which binds the mycotoxins, so we can completely say there is no concern about mycotoxins in this study. They were killed in an abattoir. Their internal organs were weighed, and then we inspected them internally. So we inspected kidneys, heart, lungs and stomach, done by 2 qualified veterinarians who were blinded. They did not know which pig was fed GM and which pig was fed non-GM, so they could not influence the results at all. And this is what it looks like in an abattoir. So the animals were led into a CO2 pit to be killed. Then they were brought along this conveyor belt. You can see the intestines here. This went off into food, ordinary US food, and we looked at the entrails. We read the entrails, if you like. And We looked inside kidneys, and we looked inside lungs, and we weren't able to look inside intestines because of this. I mean, they were fasted for a while before they came to the abattoir. But you can see, I mean, there's no hope in hell of being able to slice all of those out, wash it all out, and actually look inside. So we weren't able to see if there were any effects of the GM diet on further down the line than the stomach. But at least the veterinarians did, feel through to see if there were any tumours there, for example. So we found two main issues. First of all was that there was a 25% increase in the weight of the uterus in the female pigs. As I said, we can't... Is that on?
No.
With the microphone's died. Sorry. Is that on?
Battery life's about halfway early.
Well, I'll stand here and yell. Can everyone hear me still? Do we want this on? Just turn the... Sorry? Can we turn this one on?
Yeah, I'll just give that a go.
Testing 1 2.
Testing 1 2. Okay, can people hear me now? I will not prance around quite so much. So it's 25% higher in the GM-fed pigs, and that was statistically significant, as you can see by the p-value there. Now, there are a number of adverse things that could be happening in the uterus, and I'm happy to read out the list of scientific names if people would like. But the other thing we did then was look at stomach inflammation. And it was measured by veterinarians, one particular veterinarian, as either being nil, mild, moderate, or severe. Now, this scale was developed by the veterinarians in the days leading up to the end of this study by looking at stomachs from pigs from other farmers that had come through the same abattoir. And so, so there's the guy who was reading the stomachs. Now, you can see what he's doing. He's looking at, taking all the folds, looking underneath the folds, and what he's doing is he's counting all the ulcers, all the bleeding ulcers, the size of the ulcers, the number of erosions, the level of inflammation, all this sort of stuff according to predefined criteria. So this is a nil inflammation stomach. So that's what a healthy pig stomach looks like. If it gets too mild, oops. Right, we're back. Is that it?
Yeah.
Right, can everyone hear me? Good. All right, so this is a mild inflammation stomach here. You can see the redness is starting to increase. Then we've got a moderate inflammation. You can see the redness is increasing. And this is severe. Now, for it to be severe, almost all of the fundus has to be this bright cherry red colour. And it's not a stomach that I would like to have. So what we found is that there are, we graded every pig, or the veterinarian graded every pig. He was blinded, he didn't know which pig was fed GM or not. And what we found is that, the pigs in the United States are fed this finely ground meal. When they eat it, it irritates their stomach. And in fact, there's a view amongst veterinarians there that if the stomach of the pig's not inflamed at all, then you're not getting good feed conversion. But what we found was that the GM diet took it from the mild and moderate and pulled it up into the severe. So if you look at the severe inflammation, then overall it was 2.5 times more likely to have that severely inflamed stomach if the pig was fed the GM diet. In females it was 2.2 times, and in males it was 4 times higher. Now all the results here are the statistically significant results, because that's what one as a scientist should report. And we concluded, in fact, that it was probably likely, because you think about, these Bt proteins are designed to act on the gut of a grub, right? So therefore they're designed to cause damage to the gut of a grub. And we know that the Bt proteins, as they appear in the GM crop, have been truncated, have been changed, are synthetic compared to the naturally occurring Bt proteins. So therefore we considered that if there was a cause to this, it was most likely to be due to those Bt proteins. And in fact, it might be due to one, it might be due to the other, or it might be due to a synergistic effect of both of them acting together. The other thing we did was we measured blood biochemistry, and this is rarely done in industry studies. So we took blood 2 days before the autopsies, and we didn't find a difference between the GM and the non-GM. And that means two things. First of all, that's a good news story, as far as I'm concerned, that there was nothing that was saying, that the GM diet was causing biochemical problems, for example. But it also means that the things that you measure in blood biochemistry, which is the standard sort of blood biochemistry that you would get if you went to your doctor for a checkup. I mean, that's the sort of suite of tests we're talking about. That they're very poor measures of inflammation, because we got the inflammation in the stomach, it wasn't reflected in the blood. And it's also a poor measure of the size of the uterus, because we had a difference in the size of the uterus, wasn't picked up in the blood, and that's because the testing it doesn't pick up those sorts of things. So it doesn't pick up levels of estrogen, for example, in the blood, because that's not part of a standard biochemical test. So what we found here was that if you do a properly conducted study, a long period of time, on animals that have a similar digestive system to humans, they're fed just weaned, you control for most of the major confounders, which I can talk about more as an epidemiologist. You control for everything, that you can find these a mixed GM soy and GM maize diet cause stomach and uterine pathologies in animals. And humans have a similar digestive tract to pigs, and so the concern is that something might be similarly happening to people. Now, we've just published this study, so have I got time? Am I running out? Good. How long have I got?
About that long.
How long do you need?
No, it's all right. Oh, I've got plenty. Have I got time? Oh, cool. Excellent. Got plenty of time. Right, so this is a follow-up study. And it's a follow-up study to two things that we've done. The first one is we found the pathology, the inflammation in the stomach. Right? So we want to see, do a further study and elucidate that a little bit better. And the other thing was our histopathology study said that on these particular gene inserts that were in the diet that we fed to the pigs, there wasn't a properly conducted study or reported study. There was not a single study you could hang your hat on and go, yes, I can see exactly what they've done, I can see why they decided this was a good outcome or a bad outcome. Proper statistically conducted, the whole lot. There wasn't one of those. So we thought, we'll do one. So we'll get rats, which are the standard laboratory animal. So we've moved now from pigs to rats. Now we're not feeding soy and corn, we're just going to look at the corn. We're going to feed it to rats for long enough, and we're going to see if there's anything in the digestive system that might be obvious when you look under a microscope. And this is the study published there. So we used standard laboratory rats, as I said, we fed for 6 months. Now we used triple stack corn at 60% of the diet. Now this is what's called a semi-purified diet. This was made by a commercial diet maker that makes maybe half of all of the rat diets in Australia. We said, how high can we go? Can we go to 60% corn in the diet without upsetting the nutritional balance in the diet? And he said, yep. So we did for 60%, we made 60% of the diet. And this is also about the same amount of corn that some people in Africa eat. So it's not some pie in the sky thing we're talking about. Because we saw what we saw in pigs, we're going to look in the stomachs of these rats now and look at the lining of the stomach through ordinary and specialised microscopes, the transition electron microscope. and using different stains to look for different things. Now, there were two things that we found.
Judy, just one question. I noticed they were all males. Was
Yes.
there a reason
Oh,
why
sorry.
they were all males?
Yes, all males, because the effect we saw in male pigs was greater than the effect we saw in female pigs. Remember, it was 4 times higher? So we thought, well, we'll just concentrate on that. Well, let's elucidate. Let's see what could be happening here. So we went for male rats. Now, the first thing is we found an effect on tight junctions. Now, tight junctions are, you've got 2 cells in the body and you need them to butt up tight against each other. If they don't, if you've got a bit of a gap between them, things can leak through. So it's a bit like mortar in bricks in a wall. You don't want the breeze to flow through, you don't want water to come leaking in, and so you need that mortar. And between cells lining the stomach, these are called tight junctions. Now, to look at these, you need to use a transmission electron microscope, which is really high magnification. We're talking about 43,000 times magnification. And poor apposition should not occur in healthy stomachs. So this is a tight junction. So, like I said, this is serious magnification here. That is the junction between the 2 cells. And between the arrows is the tight junction. And you can see, this is from a non-GM fed, you can see they're butted tight up against each other. What we found with the GM, you see the blebbing that's occurring here? You see how the tight junction's blown out into a bleb? That's what we were seeing in the GM fed, and also we were seeing the complete loss of the tight junction. So you see they're apart now. So how much more commonly was this occurring in the GM fed? Every GM fed rat had poorly opposed tight junctions, and that included the blebbing, it included the complete loss of the tight junction, partial to complete loss. This poor apposition was 5 times higher in the GM fed compared to the non-GM fed, and that means it's highly statistically significant. 70% of those GM fed rats had at least 30% of their tight junctions poorly opposed, compared to none in the non-GM. And the risk of a rat having 30% or more of its tight junctions poorly opposed was 7 times higher if it was fed GM. Highly statistically significant. And in fact, 20% of GM fed rats had more than half of their tight junctions poorly opposed. So this is not a good thing. Right, I think you'll agree. Now the question is, therefore, is this causing the stomach to leak? We don't know. We don't know, because there are other layers in the stomach that would have to leak in addition to the tight junctions. So we don't know if it's a complete leaky stomach or not. That would require further experimentation. Particularly, there are tests you can do, but it would require doing a study from scratch. The other thing that we did was look at the glands in the stomach. Now, the rat stomach's interesting. It's got 2 sections to it. One half is where it stores some food, and the other part is what's called so that's the non-glandular stomach. The other part is the glandular stomach, where you find the glands. And for this, we looked at under a normal microscope, the sort of thing that you see, you think of when you think of people looking through microscopes, something that sits on the bench and you look through it. Whereas an electron microscope's a far bigger affair. Now, the stomach here has got, it's got pits, microscopic pits in the stomach. And in the pits, you've got glands that produce mucus to protect your stomach lining. And then at the bottom of that, you've got 2 glands that come down. And the glands produce hydrochloric acid to help digest your food. What we were seeing in the GM fed rats was large gland dilatation. So we were seeing swellings of the glands. So this is what a normal stomach should look like. And you probably can't see, but these are all cells lining it. And there are actually glands. So these are glands that come down and they're fairly close to each other. These are just pointing out certain particular cell types there that histologists like to look at. But you can see that everything's all tightly up against each other. That's a normal stomach gland section from a non-GM fed rat. And that's what happens if you feed a rat GM. So what we're seeing here is that these glands have now elongated, they've swollen out. So you've got a gap now between the 2 parts of the gland. And the other thing that's interesting is you probably can't see it from here, But in these particular ones up here, we're also getting what's called elongation of the cells. So in other words, the cells that are remaining have stretched out to cover the space. So if we look at the stats on this, more than 6 times as many rats had glands that were both swollen and lined with these elongated cells. So if you've got a swollen gland, that's not good. If you've got elongated glands, that's not good. You put the two of them together, that's really not good. So it's this extreme edge. It's the doubling up. It's it's the two of them together that we're looking at here. And more than 6 times as many rats had glands that were both swollen and lined. And it was often seen at the junction between the glandular part of the stomach and the non-glandular part, which makes a certain amount of sense, because if the rat's eating food and it's storing it in the non-glandular part, then this junction between the glandular and the non-glandular is where the food's likely to be sitting for the longest in the glandular area. Now, every rat fed the GM diet had at least one gland that looked like that when you looked down the microscope at that piece of tissue. No rat in the non-GM diet had a gland like that. Now, what we think might be going on is that it might be some kind of repair mechanism called restitution, so that there's some damage occurring in these glands, and that therefore the remaining cells are stretching out to be able to cover the epithelium. So we found other pathologies as well in GM-fed rats. We found dysplasia. Now, dysplasia is a bit of a worry. Sorry?
What do the glands do? Do they secrete some acid?
Yeah.
Food breakdown?
Yeah.
Okay.
Yeah, that's your stomach acid is made in these glands. So we also found dysplasia. Now, dysplasia can take different forms. The one that we were seeing was finding cells that shouldn't be here. They should be over there in a different part of the stomach, right? But they're over here when they shouldn't be. So in particular, things like the cells that produce mucus are now in the place where they should be producing acid. Now, we only saw this in GM-fed. We didn't see it in every animal, and it was only in the GM-fed. Now, dysplasia is actually, when you go for a test for cancer, like a pap smear for cervical cancer, which men wouldn't be doing, but women should be doing, as a public health person, get your smears. But what they're actually testing for is dysplasia in that instance. So it's something that may progress to cancer, it may not, it may. It may go back to being normal cells, but on the other hand, sometimes it goes and progresses to cancer, which is why we test for dysplasia and things like pap smears. There was also a 21% reduction in the number of dividing cells at the base of the pits. We don't know what that means, but we're just reporting what we saw. And if you join all of these potential adverse effects that we were measuring for and put them all into one universal scale, one global measurement, we saw that the severity of these things was 33% higher in the GM-fed rats, and that was also statistically significant. So what's happening here? Well, we think what we're seeing here, the thing that it's closest to in histopathology is chemical gastropathy, or also known as reactive gastritis, which is caused by chemical irritants like aspirin. You know, aspirin can have that effect on your stomach where they warn you, don't take too many aspirins, or if you're going to take aspirin over a long period of time, make sure it's enteric-coated aspirin. And that's because aspirin can burn your stomach. It can cause erosions of your stomach, and it can cause ulcers. So it's looking a bit like this, or alcohol can also damage the stomach so that it looks a bit like this. And there's also an organism called Helicobacter pylori, which is an organism that is being tested for a lot in ordinary people, because that can irritate the stomach and it can actually cause stomach cancer. So you can have a test. So if you've got really bad stomach pain sort of issues, it's a good idea to get tested for Helicobacter pylori, because you can be treated for it with antibiotics. We think that it's probably due to these BT proteins, because that makes the most sense, right? Because once again, they're designed to upset the stomach of grubs, and we know once again that the BT proteins that the crop produces are not the same as the naturally occurring ones, that they are all often already pre-activated, if you like, and they may have lost their species specificity, and that therefore they may be acting on the stomach. We think maybe that it might be, well, cry1AB, which is one of the proteins, one of the cry proteins, is known to bind to actin on cells, and that actin can act on tight junction proteins, so maybe that's what's happening. Maybe it's binding to the actin, that's pulling the tight junctions apart. But conclusions, we've evidenced that GM crops have harmed two species, pigs and rats. So GM crops may not be as safe as previously thought, and the Bt proteins produced by GM crops may not be as safe as previously thought. And in my view, all GM crops should undergo long-term safety testing on animals that are physiologically comparable to humans, not fish, not trout, not chickens, by independent researchers before a GM crop's commercialised, including GM crops containing stacked genes or expressing double-stranded RNA. And these tests really need to be the four major ones, which is toxicity, reproductive health, allergies, and cancer. Thank you. Oh, so I should say that if you want to, you can go to that website. I don't run that website. It's run as a fan site by other people, and I haven't looked at it for ages. But they are very kind in me posting on the website rebuttals to industry comments about it. So you'll see all sorts of rubbish written out there about the pig study and stuff, and it's all really interesting stuff, I must say. Some people have got some very interesting imaginations, but that's where you can go, and I'd like to thank you. I've got 15 minutes, haven't I?
Questions?
I've got a question.
So the symptoms that you saw in the pigs and the microscope studies you did in the rats, are they the same malfunction?
Well,
do you think?
We don't know.
I know an animal.
Yeah, we don't know because with the pigs is a different species for a start, so they might be reacting differently. The other thing is that the pigs were also fed soy, and there's a thing about soy in that it does have a bit of a reputation for making the intestinal system a bit more fragile. So that may, and also because the pig diets were finely ground. They were finely ground, so you're going to get a bit of irritation there from the diet. Whereas when you feed a rat, you feed it in pellets. So you get the corn, you grind it up the same with the GM and the non-GM, and then you add all of the other ingredients such as wheat starch and all the vitamins and minerals and stuff like that, and then it's extruded into a pellet. So the rats are eating a pellet, they're not eating finely ground stuff. So that's probably, I would think, why there's that bit of difference.
Is there any research being done using human participants? Because I'm sure the GM push, people that are doing the GM push should be volunteering.
Yes. With the lack of safety assessments and the lack of labelling, we are to a certain extent all participating in an uncontrolled trial as to whether it's safe or not. Oh, and I should point out as well, there was you were saying that saying there's only one person who's ever been killed by eating GM. As an epidemiologist, I can tell you that the two things that you did mention, which was road accidents. We've got good measurements on road accidents. We know the population of Australia. We know the number of deaths due to car accidents each year, so we can work out a good ratio for that. Also, similarly with shark attacks, but we don't know what the level of harm might be in humans because nobody's ever done a human study. So if you're going to do a human study, you need to do an epidemiological investigation, which means you need to go to sources of information to see whether they've increased over time. But that's a weak study design. You really need to go to something like a cohort study, or I'm getting too technical. But basically, there haven't been any epidemiological studies done. So we've got millions of people going to hospital, we've got millions of people dying each year, and we have no idea whether any of them, if any at all, have actually had any role, like whether GM crops have had any role to do with them. No work's been done. So you can't actually compare the number of people who might have been harmed by eating GM crops.
The risk and the process of ideals towards risk, as I said, it's
Yeah.
about beliefs.
Yes, but that, see...
And similarly, you just said, when I said oils, that cotton oil contains oil, not protein or DNA, you said, no, that's not true, because there's a peanut oil example where somebody can take an allergen. But peanut oil is often cold pressed, it's not refined because there's no gossip on it. So you've got chalk and cheese there.
Yes, but the thing also about peanut oil is...
Yes, that's a definitely. And you're right, there is no large-scale control Cochrane-type study across everybody's net. I don't think there ever would be. I think that would be far-fetched. But
Well, no, we don't need a Cochrane type of analysis. We just need to actually do some proper epidemiological studies. But the problem with an epidemiological study is that you need to know what to look for first, don't you? So do you look for... So, hold on, no, wait on. But before we go there, let's just do one thing at a time. So, if you're going to do an epidemiological study to see if GM crops have harmed people in this particular way, you first of all have to know what to look for. Do you look for bowel cancer? Do you look for allergies? Do you look for inflammatory bowel disease? What do you look for? And in order to know where to look, you first of all have to do the animal studies. So you do the animal studies first, independent, peer-reviewed science, and you go, okay, look, we think there might be a problem here. So now we're going to look in the humans. All right? Because otherwise, you've got so much to look for. And as an epidemiologist, I can tell you that we have very few surveillance systems. So therefore, it's going to be very hard to actually find out if people are being harmed.
well, it is, and we don't just eat soy and corn. We eat a whole host of things.
We do.
And we know there's a whole host of things in our diet which are often considered bad for us. I remember the kid being given burnt toast when it made nasty smells in the kitchen. And now we know that burnt toast can give you cancer. And there you go.
Well, we're talking about GMOs here rather than spreading it out more widely.
There's no GMO peanut. And it's cold pressed, so you get the allergy. So
Yeah, but hold on.
There
But
are
wait.
lines.
Yeah.
There are lines. And you're right, we need a proper epidemiological study, but
Can
we're
we,
not going to
can
get
we, before you go on, we need a bit of respect, and if the speaker's trying to answer a question, don't keep talking over. We're going to have this discussion later, and I know there'll be plenty of debate time, but let's just keep it very simple because it's really important that we learn from what we do. Okay?
So one of the reasons that people have been looking at peanut oil is because it's known to be an allergen. Right? So that's why there's been a lot of work done on peanut oil. But when it comes to actually doing a high-grade analysis of cottonseed oil as it appears in the human food supply, I would suggest to you that, we know from other work that there is a trace of protein in oils, period. Right? Because it's just, when it comes to the human food supply, it's just not that well refined. There's still traces there. So I think to state that there's no protein and no DNA in cottonseed oil is probably incorrect. However, we should do it. I mean, when it comes to things such as canola oil, for example, and to try and work out whether in safety assessments of canola oil, it would be good to test the oil on animals, right? Cottonseed oil, not been tested on animals, whether it's come from GM. And you can do safety assessments on oil. I mean, Fazant seems to think you can't possibly do it. But they're wrong, because there are thousands of items in the peer-reviewed literature about comparing olive oil to butter, where animals were fed high levels of these oils over a long period of time, and they worked out whether there was an increase in heart disease or whatever. So the work can be done. And I would suggest that the work needs to be done before you can say that it's actually safe. You need to make a decision of safety based on evidence, not on assumption.
I'm a little confused out here. I should start probably by saying years ago we heard all the studies about how they were going to genetically modify things and they were going to put a grasshopper head on a butterfly or, all this weird stuff. And that soon, flowed through the media and we were over that. And then I heard the things that were brought up earlier about the time for GM modified plants, that they could do this so much quicker than the old-fashioned breeding of, plant-based breeding and crossbreeding and everything. And that all seemed good to me. And only about a year ago, I said to some friends who are permaculture farmers, I said, well, I don't know, this GM seems fine to me. It just seems to be a speeded up process of crossbreeding. Then we went and listened to the Rockhampton Healthy Soils, and Dr. Huber was speaking at that event. And all of a sudden it became clear to me that I needed to ask, was there any other reason that they genetically modified plants other than Roundup And the answer was just the two. That was for glyphosate or Roundup Ready plants and SBT. And I sort of then became focused on the fact that and Dr. Knox's question about are you against GMOs or not? And my answer was, I don't want to go there. I don't like them. But that was based on the reason that they were built. And now I listen this morning and find out that it takes about the same amount of time to do either a GM plant or a regular old-fashioned crossbreeding. And also Professor Huber's input about the lack of variety in the plants, like the Williams 12 variety of wheat is now predominantly, and we run the risk of in alfalfa or lucerne and wheat of other things coming in. And I was still thinking that it was the reason of the Roundup, because I'm thinking, like a lot of people, that Roundup is not good. But am I understanding now that in the BTs, and I hadn't focused on that before, that your studies indicate that it was the genetically modified with the BT in it that was causing the stomach problems?
Yes. That's the most likely contender for us, yes.
Okay, and you don't think that the soy in the diet with the estrogen content, xenoestrogens, was a part of that?
I'm sorry, are we talking about rats or pigs now?
Well, I'm going both, but in the pigs you had that increased uterus size. Do you think that there's any influence from the diet of the soy?
So let me just answer that one first, if that's right.
Yeah.
And that is, we don't know. But the soy, so the pigs were fed a mixture of GM soy and GM corn, and the control ones were fed a mixture of non-GM soy and non-GM corn. So if there's an increase in the estrogenic capacity, or estrogen component of the soy, it's associated with the GM soy, you see.
Okay, so it was the same soy in each diet. So
It was the same amount of soy in each.
Okay.
Yeah, it was the same amount of soy in each diet, just that one had GM soy, one had non-GM.
Yeah, good. I missed that. So, and I assume
and then in
that,
the rats, there was no soy there.
Yeah,
It was just the
feed that would have the genetically modified for Roundup Ready or whatever, that had never been processed. It was never had seen Roundup. That's correct.
Oh no. So when we fed it to pigs, that diet had soy in it, and that came from farmers' fields as it would appear in the human diet.
So it might have been sprayed.
Oh, I imagine it would have been, yes. When the industry tested Roundup Ready soy, they tested the soy and they didn't spray it with Roundup. So it was not the same as you would eat in your diet. It was unsprayed soy. When the crop is designed to be sprayed with soy.
Because I'm still having trouble trying to figure out how much is contributed to how the plant is genetically modified, given the circle up there, and how it's put in and all that, with the chemical that's put on.
And a component of glyphosate.
Yeah.
So you're trying to I gather what you're saying is you're trying to work out what component the fact that it might have been sprayed with glyphosate has compared to the GM crop as such, the actual
The virgin
plant.
plant.
Yes. We don't know. The thing about GM crops that are designed to be herbicide tolerant, though, is that the herbicide and the crop are generally joined together. So they come together as Siamese twins, if you like, joined at the hip. So I don't know, I'm not aware of this work because I'm not a specialist in glyphosate, but there is a view out there that the Roundup Ready crops have higher levels of glyphosate in them because they can be sprayed while they're growing. But I don't know, I haven't seen the results of that, I don't know.
So Judy, basically, the results that we got there, right? We don't know how much the glyphosate's contributing to this, or how much the GM.
No, we don't. We don't know what the levels of glyphosate were in these feeds. But I point out that we've done 2 studies, and it would be unlikely we would have had exactly the same levels of glyphosate in both studies. The other thing, of course, is that non-GM crops can be sprayed with glyphosate. There's been an increase in people spraying their crops as a desiccant so that they can harvest a little bit earlier or get their or to one stage so that they can harvest cleanly. So that's going on as well. So while there may be, some argue there would be higher levels of glyphosate in the GM, it may not be the case because of the extra spraying that's occurring with glyphosate. And we're also getting considerable glyphosate-affected soils in Australia where glyphosate, and we've got a farmer here who can attest to that, where the glyphosate goes into the soil, doesn't break down as fast as the company would like you to believe it does, and you can end up with considerable residues in the soil remaining. So then if you plant a conventional crop there, you might be getting glyphosate coming up into the plant, but wasn't actually sprayed on it. So would you like to add to that? Glyphosate in your soil?
Well, glyphosate has got residual, and it is building up in the soils. A lot of the farmers have been using it for 20 years or more, and GRDC has done studies through all the states, not so much in Queensland, where they checked the residual of chemicals in the soil. So, and what they found, they checked all the chemicals, and what they found is that they found glyphosate everywhere where it was being used. The only place they didn't find it is if you didn't spray it in the paddock. And they checked from 0 to 10 and 10 to 30. And everywhere where they were spraying it, it's got past the 10 and into the next layer. So, glyphosate has got residual, it is building up in the soil, and they've got, the figures are there. So,
But they haven't released them, have they?
no.
Well, he knows about it, so he must have.
No.
No, we know about it.
They tested on his property.
I've got some testing done, I'm in CQ here, and I've got 3 paddocks done, and we've got high levels of glyphosate in our soil because we've been using it for 20 years. And it's worse in Western Australia and South Australia and sandy soils. Heavy clay soils got the ability to buffer the chemical. So there's more problems in South Australia and Western Australia and in the light soils. But farmers are ringing up scientists that have been working on this and saying, what's wrong with your crop? Because when the glyphosate levels get to a certain level, they're going to do damage to the crop. So, it is a real issue, and how it all come about through the soil biology. They had the Soil Biology Initiative 1 and the Soil Biology Initiative 2 program, where they spent 17 million, and those results were finished in 2015, and a lot of things came out of that. And the farmers wanted to know what the chemicals were doing to the biology in the soil, so they continued with more studies, and they checked all the chemicals because they got the technology to do that now.
Do you know which tests they're using? Because I grow a Roundup ready crop, and I do soil tests, and we do chemical residues all the time because we're in risk catchment. And I've never had Roundup, I've never had glyphosate picked up ever. Do you know what test they are using?
Well, quite possibly.
Testing not much good.
What they're called.
Yeah,
It's quite expensive
what
test.
method of testing are they using?
Well, the proper ones that they can pick up.
Yeah, it's good to know. The reason I would ask that question is, well, our work currently has us on Quarry Island, way down in the south, and also on some of the islands off Newcastle and Port Stephen and stuff, where we're looking, we've eradicated the bats and rats, and now we've got a problem with the grass seeds and everything else we've left in there. And one of the things we're looking at there is actually measuring glyphosate residues to work out what it is that doesn't in these systems. And the biggest problem we have is the standardised method has such huge variability that even in response to dosing standards and measurements within an hour in the lab, you get very variable kind of outcomes. So it'd be good to know who's done that and how they've done it. The GRDC method.
Yeah, well, I can give you all the information.
That's brilliant, yeah.
Yeah, no problem.
It was done by a government department.
They were checking for all the chemicals.
GRDC.
You know, Roundup was the one that we were interested in because I was a bit like Renee with, you know, the residual type chemicals. I've been reluctant to use them and thinking that glyphosate was, you know, my better option. And you know, so we actually got some testing done. But anyway, so, you know, I was talking about, yeah, they did extra work. The farmers wanted to know what, you know, what the chemicals are doing to the biology in the soil, and what they found out was that, you know, the glyphosate was being picked up and was in all the samples that they checked in you know, where they'd been spraying it. I was going down to see my daughter in Canberra, and I just picked up the Land magazine, Land newspaper in New South Wales at the service station, and they had a half-page article. And that was just after the 2000, I've got to get the years right now. I think it was 2016 it might have been, GRDC update in Goondiwindi. So the scientists involved presented at the GRDC update in Goondiwindi, and must have been in 2016. 2017 there was nothing said. 2018 there was nothing said. So everything went really quiet.
So have they got quiet with the soil biology phase 2 closing, or is this actually done at the Northern Panel and the restructures GRDC's thrown at somewhere, you think? Or because the soil biology stuff that's done in those first 2 rounds has actually moved to a Western Australian website, so it looks like it's all in WA, but it's actually everybody's information is there.
Yeah, this wasn't part of soil biology too. It was actually separate projects altogether. And it was actually run by Dr. Nick Rose and
Lucas Fang.
Okay. And they actually had the papers presented to GRDC 2 years ago, and nothing has come out yet.
So Nick Rose and Lucas
I've
Fang
been Nick for 15 years, I'll give my mind.
Well, he's probably been told to shut up. And we've got no problem with saying that, because I think it's very serious that they withhold this information from growers when it should actually be in the public arena.
Yeah, I agree with that.
So what they found out, so you had that soil biology initiative 2 work, and they've done a lot more work since then, but that information hasn't, well, they won't talk to us anymore.
That's been
They're
released to us.
probably scared of losing their job or something, I don't know. But you
As much as leverage. I don't know how much you agree to the board acre, but do you have any leverage with the GRDC Northern Panel?
Not
well,
anyway.
No.
I got up at a meeting not long ago and I said, well, the soil biology 2 information, I've been waiting since 2015 and I got it the other day, but you know, I'm the only one that's got it. So that's not good.
Thanks, Tony. We'll keep going. Is there any question for
Everyone.
question for everyone? What's the question?
Well, whether you go Roundup or whether you go GM, there's information out there now that says that even hybridising things, even pasteurising milk, even lock feeding cattle, turns a good product, a health product, into a poison to it. So how far do we have to go before we say no?
That's a good question. We might answer it this afternoon. Thanks, Berg. Thanks, Judy. Yeah, the next session was really